I'm melchore and I will be your instructor in this SID online class entitled The Wonder of Science. In this SID class, we will be focusing on the cell. Okay, let's define the word cell. Cells are the basic building blocks of living things. The human body is composed of trillions of cells. They provide structure for the body, take in nutrients from food, convert those nutrients into energy, and carry out specialized function. Cells also contains the body's hereditary materials and can make copies of themselves. Cells have many parts, each with a different function. Some of these parts, called organelles, are specialized structure that performs certain tasks within the cells. As we go on further, we have to take first the history of the cell. Okay? First, the history of the cell. We have here first Robert Hooke. In 1665, a British scientist named Robert Hooke observed mass of tiny cavities from the thin slice of cork. With his self-made microscope, he named this structure cells, since this structure reminded him of the small rooms in a monastery. So, Robert Hooke observed cell in a thin slice of a cork, and he named it as a cell. Okay, next on line is we have Anton van Leeuwenhoek. In 1674, a Dutch scientist named Anton van Leeuwenhoek made pioneering discoveries concerning protozoa, red blood cells, capillary systems, and the life cycles of insects. He also perfected the construction of the compound microscope. So Anton van Leeuwenhoek developed a compound microscope. Next is Robert Brown. Robert Brown, a British botanist, observed plant cells within a distinct central parts, or what we call nowadays as a nucleus. Described the streaming movement of the cytoplasm, or what we call on science as Brownian movement. The other one is Dujardin. Dujardin, in 1835, a French biologist, observed that cells were not empty, but filled with thick, jelly-like fluid, or what we call the protoplasm. He described the protoplasm as a thick jelly-like. And that's the work of Dujardin. And now we have here Matthias Schleiden. Matthias Schleiden is in 1838, a German botanist, concluded that plants are composed of cells and formulated the plant cell theory. So Matthias Schleiden formulated the plant cell theory. We have Theodor Schwann. 1839, German zoologist Theodor Schwann concluded that animals are composed of cells and formulated the animal theory. So Theodor Schwann formulated the animal theory. And then next we have Rudolf Virchow. A German pathologist concluded that all cells must come only from pre-existing cells. So Rudolf Virchow explained that all cells comes in one cells into another cells. Next online we have Max Knoll and Ernest Ruska. A German engineers built the first transmission electron microscope. Then we have James Watson. James Watson, an American biochemist, and Francis Crick, a British biophysics, discovered the structure of DNA that ushered in the era of molecular biology. And they form a theory, what they call cell theory. The cell theory is the observation and conclusion of Schleiden, Schwann, and Virchow that they have two objectives, which number one is all organisms are made up of cells, and the cells is the structural and functional unit of organisms. Second, cells are capable of cell reproduction, and cells come only from pre-existing cells. Now, we will go to the most important part of cells, the functions and the parts of the organelles. Okay? First, we have the plasma membrane. The plasma membrane or cell membrane is what they call the city border. The function of it gives strength, shape, and protection to the cell. It also regulates the traffic flow of materials between the interior of the cell and its environment. So the plasma membrane, also called the city border. Next is the cytoplasm. This part is the cell content outside the nucleus. The ground substances of the cell composed of protein, carbohydrates, and lipids dispersed in a watery medium resulting in a colloidal solution. It also the most active region of the cell due to numerous biological and biochemical reactions of the organelles and its inclusion. Cytoplasm is always called the lungs or the part. Okay? Next is the nucleus. The most distinct, usually situated at the center of the cell, bounded by a double membrane, or what they call a nuclear membrane. Nucleus controls and regulates the function of the organelles. Thus, this nucleus also called the governor of the cell. On a nucleus, it contains nucleoplasm, then nucleoplasm, nucleolus, and chromatin materials. The nucleoplasm or the karyolymph, the colloidal solution enclosed by a nuclear membrane, more viscous than cytoplasm. Next is the nucleolus, a counterpart of ribosomes in the cytoplasm for protein synthesis. The other one is chromatin. Material contains the linear arrangement of the basic unit of heredity called genes. We have the endoplasmic reticulum. The endoplasmic reticulum forms the cytoplasmic skeleton composed of complex system of membrane. The endoplasmic reticulum serves as routes for the transport of material between the various parts of the cytoplasm. The endoplasmic reticulum, we have the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The rough endoplasmic reticulum is the source of protein, while the smooth endoplasmic reticulum is a lipid synthesis. It also processes a variety of metabolic process such as drug detoxification. Next online, we have ribosomes. The ribosomes are spherical bodies that may attach to the ER or free in the cytoplasm. Aggregates of RNA and protein which serve as a site of protein synthesis in the cell. Proteins synthesized from free ribosomes are released as enzymes. Proteins from attached ribosomes are for the use with the cell. The next organelle was the Golgi apparatus. Golgi apparatus is a membrane-bounded vesicles of flattened stalks and stalks parallel to each other. Serves as the storage, modification, and packaging of the secretory product in vesicles. Golgi apparatus is the shipping goods created by the endoplasmic reticulum and ribosomes to the rest of the cells. This is what we call, they are the post office of the cell. Now on deck we have mitochondria. Mitochondria is a double membrane organelle. It has a smooth outer membrane and folded inner membrane, or what they call cristae. Mitochondria is also the site of cellular aerobic respiration, and in chemistry they call it Krebs cycle. It is commonly called the powerhouse of the cell, or the energy plant of the cell. We have here lysosomes. The function of the lysosomes is to break foreign particles of the cell, or may break down, or what they call autophagy. It is a proteinaceous component of the cell itself by destroying organelles. Lysosomes contains of hydrolases, which catalyzes the digestion of proteins, nucleic acid, and some carbohydrates and fats. This will be our final discussion. And for your evaluation, I want you to answer on the following slides on Padlet that I have given unto you. So, take a journey, enjoy, have a safe trip.